Hi, I'm Bernice Kier,
And my name is Mary Nethery.
and today we will be presenting on the enzyme Phosphfructokinase-2. So let's talk about the enzyme. So the enzyme is a member of the ribokinase family. It is a bifunctional enzyme. It has two domains, a kinase domain and as well as a phosphatase domain. It is a homodimer, so as you can see here, it has two parts that are the same, hence the name. In its active form, it is the homodimer, but when it is inactivated by the macromolecule ATP, it forms a tetramer, and in the tetramer form, that is when it is inactive and cannot perform its functions in the body.
So the main purpose of this enzyme is to regulate glycolysis and gluconeogenesis. Glycolysis is when one molecule of glucose is converted into two molecules of pyruvate, and during this process ATP is generated. Gluconeogenesis is the opposite of this process, where glucose is generated from two molecules of pyruvate and ATP is consumed. Phosphfructokinase-2 regulates both of these processes depending on its phosphorylation state. When it is dephosphorylated, it is deactivated, it acts as a kinase, and glycolysis is activated. When it is phosphorylated, it is activated, it acts as a phosphatase, and gluconeogenesis is inhibited.
Here we have the primary sequence of our enzyme. So as you can see here, towards the C-terminus of our enzyme, towards the N-terminus of our enzyme, we have a lot of beta sheets which are represented by the yellow arrows. Whereas here, towards the C-terminus of the amino acid sequence, you have a lot more alpha helices represented again by the red squiggly lines. In the paper in which we're going to present today focuses highly on the C-terminus end of our enzyme, and that will become more relevant as we continue our presentation.
So the primary literature that we're discussing for this project is the uncoupling of MG-ATP-induced inhibition and aggregation of Escherichia coli phosphofructokinase-2 by C-terminal mutations. A little bit of background information for this project. In previous experiments, they were able to get fragments of the enzyme via proteolysis, and these fragments were able to retain their allosteric binding capability, but they weren't able to form tetramers. This led them to believe that the C-terminus was responsible for tetramerization, and it also led them to believe that tetramerization and the allosteric binding were independent of one another, but they weren't sure which region of the enzyme was responsible for both of these processes.
So the goal of this paper is to dive into and explore the structural determinants that basically govern the ATP binding sites, as well as the structural determinants of the ATP-induced tetramer conversion.
So for the first part of the methods and materials of this paper was generating the mutants for this project, mutaneogenesis. They did this by inserting stop codons in the C-terminal region of the enzyme. This therefore cut off additional amino acid residues towards the end of the enzyme and therefore generated the mutants. It's also important to note that the D299 mutant could not be purified in this experiment, and therefore they had to adjust the KCAT values accordingly.
The second part of the methods was size exclusion chromatography. Once the mutants had been created by way of mutaneogenesis, size exclusion chromatography was the technique used to separate them, again by size. Here we have the ladder, the macromolecules that are composed of the ladder that were used to separate our mutants. And here we have a image which displays our 4 mutants here, and so you have a total of 4 mutations, 2 to 10 deletions. And here the first one represents the wild type, where you have all of your amino acids present. The L307 is the first mutant with only 2 amino acids gone. The second is Y306 with 3 amino acids gone. The third being A305 with 4 amino acids, and the last is D299 with a total of 10 amino acid deletions.
So the third part of this methods and materials were the enzyme assays. The activity of our enzyme phosphofructokinase-2 was determined using a spectrophotometer, and the Km values for the MGATP were calculated using the uncompetitive substrate inhibition function. And for the fructose 6-phosphate, they were calculated using a nonlinear curve fit to a hyperbolic function.
So for the results, here the first part of the results, we have again the image of our wild type and the mutants just for sake of reference. The main results from the first part of the experiment showed that while the Km value, which measures affinity for substrate to enzyme for the fructose 6-phosphate for the PFK2, essentially did not suffer any severe changes, the Km values for the macromolecule ATP, MGATP, increased significantly as the number of amino acids removed from the original sequence also increased. So we see here as well the kcat values remained relatively unchanged.
These two graphs here show the activity of the mutants versus the MGATP concentration. We have the two mutants, Y306 and A305, and we also have 3 different concentrations of fructose 6-phosphate. As you can see, the enzyme has a maximum activity, and then as you continue adding MGATP, the activity decreases. This suggests that the allosteric inhibition is still functional even though the Km values for the mutants was around 1.5 times higher than that of the wild type.
Here we have size exclusion chromatography that was performed in a rather low concentration of our one of our substrates, the ATP. So the dashed line represents no substrate, and the continuous line represents, again, the low concentration of our ATP. You can see here the wild type enzyme, you can see when there's no substrate there, compared to when there is substrate present, there's a significant change in the RH value. The only mutant that exhibited a similar change in the RH value was the L307, which only had a total of 2 amino acids removed from its sequence. The other 3 mutants, from anywhere from 3 to 4 to 10 deletions, did not show a change in the RH values. This lack of change indicates that there was no tetramization, although previously in the slides, looking at the kcat values, we still know that there was a reaction occurring. There was still allosteric inhibition occurring, by the ATP molecule on our enzyme.
So this chart here introduces 2 additional mutants that were generated in this experiment, the Y306A and the L307A. These 2 mutants were generated by a point mutation, and one of the side chains on each mutant was replaced with an alanine. This table here shows that the Km for the MGATP for the L307A mutant was much higher than the wild type, while the kcat and Km for the fructose 6-phosphate remained essentially unchanged. This diagram here shows the dependence of the initial velocity on the MGATP concentrations. It shows 2 different concentrations of fructose 6-phosphate, and it shows both mutants as well as the wild type. As you can see for the L307 mutant, the peak activity is at much higher concentrations of the enzyme, and this is due to a pseudo-affinity that this mutant had. From these graphs, we can see that the C-terminal deletion mutations were not able to form tetramers in the presence of ATP.
Here we have an image that was created from the size exclusion column. We have the dashed line, which represents the wild type enzyme, and our continuous line is our Y306A mutant. You can see here that at high concentrations of our substrate MGATP, 0.8 millimolar, we have this RH value for our wild type, whereas at the same concentration of our substrate ATP, the Y306A mutant does not show a similar RH value. This indicates that even at high concentrations of the ATP, which is the allosteric inhibitor of PFK2, the Y306A does not undergo that dimer to tetramer conversion. So this indicates that the side chains on the Y306 are crucial for this conversion, but they are not required for the ATP binding to the active site of an enzyme, because as we saw earlier, our KCAT values, they were similar. Even in the previous slide, the KCAT values are still standing to indicate that there is indeed a reaction occurring.
So for conclusions, this experiment was kind of broken up into two parts. So for the kinetics part, they found that the mutants L307, Y306, and L307A all had an increased Km value for the MgATP, but had no significant changes in the Kms for the fructose 6-phosphate. Of course, an increase in the Km is a lower affinity, and in this image here that you can see from the RSCB protein data bank, we can see the ATP inside of our enzyme in its native form. You can see that there are a lot of hydrophobic interactions taking place between the ATP and the enzyme, and these interactions would not be there after the mutants were created. This is because the mutants that were created are known as cavity-creating mutants, and therefore it affects the tertiary binding packing around the ATP, and these hydrophobic interactions are therefore lost, which is why the affinity is lower.
In regards to the formation of the tetramer form, it was believed that the dimer to tetramer conversion was directly linked to the ATP molecule binding to the PFK2 enzyme, thus inhibiting it. The results of this paper and this experiment indicate that the two processes are not necessarily dependent upon each other, that they occur simultaneously. So while you may not have the dimer to tetramer conversion, you can still have the binding of ATP to the phosphofructokinase-2, as well as the allosteric inhibition of the enzyme by the ATP.
And these are our references, and all of the pictures and the primary sequence for our enzyme were all obtained from the RSCB Protein Data Bank Bioinformatics website.
Thank you for watching.
Thank you.
Bye.